Good morning, and I will give the lectures on the female gonads. So the first one is on the ovary and uterine tube. So ovary is a complicated slide and a complicated topic. Here we will talk about follicular development. Half of it you already know, if not all of it from last semester. So it is complicated, but half of it is revision. We have to also review and learn new things about the cyclic hormone secretion. These are the two main things that happen in the ovary. Parts of the ovary, now of course we're focusing on histology, but you will learn it soon in anatomy as well, and you will see and be able to connect the macroscopical parts with the microscopical parts. The cortex is the outside, of course, as in almost all organs. The cortex is outside, and this is the main part. It's the biggest part. And here inside, we have the different follicles. I choose another pointer, different staged follicles. So that's where we have the follicular development. And the follicles are embedded in the so-called stroma, which is the framework tissue. It's the so-called spinocellular or cell-rich connective tissue. We only mentioned it last semester when we learned the main types of connective tissue, but we haven't had a slide yet. So this is the first time we do see the spinocellular connective tissue, a very cell-rich connective tissue. I will show you with higher magnification that it's really, really cell-rich. And near the hilus, this is the hilus from where the blood vessels reach the ovary. Blood vessels reach it in the medulla and then divide in the so-called medulla. And of course, along with nerves and some endocrine glands, which are found inside this loose connective tissue, which fills the medullary part. The ovary has a very good blood supply, so rich blood vessels are found here, big blood vessels. Also in our slide, you can see big blood vessels. So the surface, let's continue with the surface. The surface is partially covered by peritoneum. Now you are doing the abdominal cavity, so you know about the peritoneum, about these anything. So mesentery, mesogastrium, etc., always indicates the double-layered peritoneum that goes to the organ to surround it. The ovary has a very special relation to the peritoneum. It is not intra, not extra, and not retroperitoneal. It's somewhere in between, because as you can see in this drawing, or if you put your finger through a buttonhole, you can actually understand this relationship. So if you do like this, you understand how the ovary is related to the peritoneum. There is a double layer of peritoneum going to the ovary. So it has a so-called mesovarium, which is, of course, peritoneum. So histologically, it is mesothelium. And it covers partially the ovary, but then it stops. This is simple squamous epithelium or mesothelium, but the bigger part is not covered by it. As if it is looking through a buttonhole into the peritoneal cavity. So it is not covered there by mesothelium, but is covered by simple cuboidal epithelium, which is called germinal epithelium. This is what it is called, but the origin of the word is on a wrong conception, because scientists thought that from this epithelium, this is where the oocytes are from. So, of course, it's a wrong conception. They are not coming from this epithelium. That's why it was called germinal. It has nothing to do with the germ cells, but the name has stuck. So this is what it is called. Later, you will see that it's a very important surface. In pathology, you will learn about ovarian cancer, but everybody heard about ovarian cancer. Unfortunately, a quite common type of cancer and really, really malignant. And most of the times it comes from this surface epithelium, so the germinal epithelium. And the border between the two lines or the two coverings is called the Farey line or line of Farey, which you can see macroscopically. So in anatomy, you will be able to see it very well. Okay, let's continue with what we have under this epithelium. So here you can see the so-called Tunica albuginea. It's a denser type of connective tissue. And when you do the testis later, the male genital organ, then you will see a real, very thick, macroscopically really white adipose tissue, white connective tissue. That's why it's called Tunica albuginea. In the ovary, it's much thinner, but it has the name Tunica albuginea. And then comes the stroma, where we have all these follicles embedded here. And you can see the surrounding connective tissue, which I mentioned in the beginning. This is the so-called stroma or spinocellular connective tissue. Here you can see how rich in cells this connective tissue is. And embedded, we find the different follicles. So let's see the follicular development. Half of it will be reviewing what you did last semester in embryology. So first of all, let's see what a follicle is. Sometimes people are not aware what, they use the word, but they are not aware what exactly follicle means. Follicle is the oocyte plus the epithelium or later the Theca cells surrounding it. So this whole structure is the follicle. And now we have to review a little bit oogenesis. And you had several questions actually regarding the follicular development and ovulation. So all these topics you should review from last semester embryology. So I'm going to use Dr. Horvat's first semester slides, so you can recall better that you have seen these already. So oogenesis starts, as you know, during embryonic life already. The germ cells migrate to the genital ridges from the yolk sac and then differentiate into oogonia and then undergo mitosis. And they reach several millions in number during embryonic life. And then already in embryonic life, the number is going to decrease. And when we are born, it's between 1 and 2 million. And then during childhood, it further decreases till puberty, when actually oogenesis will continue. Maybe you remember these, hopefully you remember these graphs, and you remember the number is continuously decreasing. And this was oogenesis, this was one entire topic in the first semester with all these numbers that you had to learn. And there are several important differences between oogenesis and spermatogenesis. So as I said, oogenesis starts in embryonic life, but then will be stopped. All the oogonia will turn into primary oocytes with 46,4N. And then, thanks to this inhibiting factor, the meiosis inhibiting factor MIF, the further development will stop. So this is the end of prophase, and the development stops here. Until puberty, actually no further development will happen. This whole thing lasts about 6-7 months. I will show you further graphs and emphasize further that it's not 2 weeks. Because after puberty, when this whole thing continues, from puberty till menopause, of course, then every month we have one follicle, one oocyte that reaches full maturity. But the whole process is not 1 month or 2 weeks. the whole process is 6-7 months. So after puberty, or at puberty, the thing continues, and then first meiotic division leads to the secondary oocyte, which has 23-N, and the secondary oocyte is only happening once a month before ovulation, is when first meiotic division is completed. And then the secondary oocyte starts the second meiotic division, and the mature oocyte is we only have mature oocytes when fertilization happens. So 23-1N, the haploid chromosome number. This is still revision, the follicular development. So not only the oocyte, but the whole follicle. This is what you did already in first semester, but now we're going to do a little bit more details. So this is what is a little bit new already, the follicular development. We have different categories and the names differ between sources. So if you open 3 books, maybe you find 3 different categories, and actually all of them are used. So it's good to remember these 2, okay? We have, of course, the primordial. It's the same in every grouping, primordial follicle. And then we can have, as you see, with the growing epithelial layers, we can have the so-called pre-antral before the antrum appears. So we have pre-antral phases and antral phases. But I think the best to remember is this, that we have primary, secondary, tertiary. It's also easy to remember. But both are very, very useful. So here are the details. So again, showing you both types. So we have the pre-antral phases before the antrum, before this fluid appears, and the antral phases with early and late antral phases. The primordial follicle here has squamous epithelium, simple squamous epithelium. We have, of course, most follicles in this stage, also in the slide. And as you see, during the follicular development, the size really grows of the whole follicle. Actually, this is so big that if we wanted to do it in proportion, this wouldn't fit in the picture, because as you see, from 20 to 40 micrometers, the Graafian follicle reaches 15 to 20 millimeters. It means 1 or 2 centimeters. So it's a macroscopically visible big thing. So all these drawings are not proportional. And during this growth, even the oocyte is growing. You see, from 20 to more than 100 micrometers, the oocyte itself. And so if we go back to the primordial, simple squamous epithelium surrounds it. Then it starts to grow. First, simple cuboidal. This is the primary follicle. Then many layers of the cuboidal epithelium. This is the secondary follicle. And this stratified epithelial layer is called granulosa cells. So from this point, we call them granulosa cells. And at this stage, also from the stroma, the Theca cells will differentiate. So Theca cells also appear around the granulosa cells, the Theca interna and Theca externa. And then when this further grows and many layers develop, then we have a cavity filled with fluid. First, a small one appears, then several, and then they fuse to give a huge antrum. This is the early or immature and the late or mature Graafian follicle, also called tertiary follicle. And here, where the oocyte remains surrounded by the granulosa cells, this is called corona radiata, and cumulus oophorus is the area, the island, the islet where the oocyte is embedded. Be careful. I made a red square around it. It can be a tricky question. We don't ask it because it's a tricky question, but it can be a tricky question. You see the follicle is called primary, secondary, tertiary. And if we ask in what stage is the oocyte, the oocyte inside the follicle is always primary. If you remember going back to this picture, the primary oocyte finishes the first meiotic division and becomes secondary oocyte only before ovulation, shortly before ovulation. So it means that all the oocyte, except for one maybe that is going to ovulate now, so all the oocytes are primary oocytes, okay? So this is very important. You will learn later in gynecology and also in physiology, it's even more detailed how this whole follicular development is happening. And just remember the dates that we have the pre-antral growth, so from primary, from primordial to pre-antral follicles. This takes the longest time, for 5 months. Then from pre-antral to small antral follicles, 2 more months. And then the exponential growth phase from antral to Graafian, the biggest antral follicle, is again 3 weeks approximately. Now comes a very important slide, and you have to really practice yourself all these graphs to draw. Please do it. When we have normal lectures, we always do it together. Do it in class, do it at home, and learn the hormones very well. This part, we don't need it so much in detail this semester, because next semester we will learn about the hypothalamus in detail and the pituitary gland in detail. But you do have to know that above all the hormones stand the hypothalamus with the gonadotrope releasing hormone, GnRH. This regulates, so this is the main boss of the endocrine glands. This regulates the pituitary gland. This, you do have to know these two hormones, the FSH and LH. FSH, the name says follicle stimulating hormone. So this stimulates the follicular development that we just described. And LH is the luteinizing hormone. Okay, this is the one that induces ovulation. This was already a topic last semester in embryology. And the development of the corpus luteum, this is why it's called luteinizing hormone, because it induces the development of the corpus luteum. So this is how these two hormones change during the cycle. As you see, and it's very easy, the LH has a peak shortly before ovulation. Shortly before ovulation. Please remember this graph. Many graphs that you find on the internet are wrong because they put LH peak to the middle of the cycle, and it has to precede at least 1 day, so at least 24 hours, the ovulation, because this is the one that induces ovulation. Okay, so this graph here is correct. Here you can see the menstrual cycle that you already learned in the first semester. So if we consider the average 28 days, ovulation is in the middle, is on day 14, and the LH peak precedes this. And then it goes down again, okay? So that's easy. FSH has a smaller peak at the same time, but you see it goes up and then down. Why is this up and down and again a peak? Because in the beginning, the follicles grow independently. I will mention it again later. Will grow independently of all these gonadotropin effects. So this is called the gonadotropin independent phase, when we have few FSH receptors. So there is estrogen produced by the developing follicles, and there is negative feedback. So first it goes up, and then because of the negative feedback, it goes down. And then as FSH decreases, only the dominant, most sensitive follicle will survive, and this is the Graafian follicle. The others become atretic, and then we have a positive feedback. Details of this you learn in physiology, and later this in anatomy in the 3rd semester. Okay, just remember this: first negative, then positive feedback, and we have another little peak here. So follicle stimulating hormone stimulates the development of the follicles in the gonadotropin dependent phase. And then the follicles produce estrogen. Estrogen, here you can see the 2 hormones produced in the ovary, estrogen and progesterone. Okay, and here you can see estrogen increases because of the developing follicles, and it has a peak also before ovulation, but it's also present during the 2nd half of the menstrual cycle. Progesterone, on the other hand, is only produced in significant amount during the 2nd half by the corpus luteum. You can see 1st half almost nothing, and the 2nd half is when it is produced in increasing amounts. And these 2, the estrogen and progesterone, will affect the uterus. Next lecture, I will talk about this in detail. And the whole point of this cycle is to prepare the uterus every month for pregnancy. So this is the whole point, and you have to know these hormones very well for not only the ovary slides, but all the uterus, the mammary gland, etc. slides, because we will ask these hormones always, like uterus, progesterone has the progesterone phase, induces the progesterone phase of the uterus, where is progesterone produced, etc. So you have to know this very well. So let's see the population of the developing follicles. Of course, most follicles are resting follicles. They wait for the next month to develop, or whenever they're going to develop. And then we have a group of follicles that begin to grow. We don't know why and how a certain group of follicles is chosen, but or selected. And this is the gonadotropin independent growth. We don't need those hormones for this growth, okay? To an early tertiary phase, this happens. This is the gonadotropin independent growth, which happens even in children before puberty, but of course, these follicles will degenerate. And then we have the gonadotropin dependent phase, when again, a group of these developing follicles are recruited. This is called recruitment of these follicles from the tertiary follicles, and only one of them will be the dominant follicle, which is the Graafian follicle. Of course, in case of twins, there can be more than one, but usually one reaches full maturity. The hormones produced, I already mentioned the estrogen and progesterone produced by the follicles. The before corpus luteum, we had estrogen production, and this is a cooperation between Theca interna and the Granulosa cells. So the Theca cells produce the androgen, and then it's given actually to the Granulosa cells. Here you can see this biochemical reaction from androstenedione. The aromatase enzyme present in the Granulosa cells makes the estradiol. So estradiol is the main estrogen produced in the ovary. This whole process is stimulated by FSH, mainly FSH. And we have, of course, several other hormones. Some names tell you what they do, like inhibin inhibits FSH, like it's a negative feedback, or activin activates FSH. Relaxin relaxes the uterus, so the names are also helping you to find out what their function is. And we have one hormone that I encircled here, the anti-Mullerian hormone, which is used already in tests, in laboratory tests, and it's missing from most books, most textbooks. So that's why please learn it, and that's why I emphasize it so much. You will learn about the anti-Mullerian hormone when you do the development of the male genital organs, because male genital organs develop from the Wolffian duct, female genital organs develop from the Mullerian duct. You will learn it later this semester. So the anti-Mullerian hormone inhibits the development of the Mullerian duct, so it stimulates male development. So it's present in here. It's produced by the Sertoli cells in male fetus. And in females, it's present mainly after puberty, in reproductive age, and it decreases sharply before menopause, and then almost completely disappears, because it correlates. It's produced by the follicles, and it correlates with the number of follicles. So it's used to estimate the follicle number, how good the ovarian reserve capacity is. So that's why it's used already as a laboratory test. That's why you should really know about it. So anti-Mullerian hormone. Now let's see a little bit more details about the histological structures. Here, the oocyte is surrounded by the Zona pellucida that you already learned in embryology as well. Here you can see it as an eosinophilic line. It's very obviously visible. And we have the Zona pellucida and corona radiata, then the Granulosa cells. They are very important during when the sperm comes and for reproduction. And you learned this also in the first semester, that there is the so-called acrosome reaction, when the acrosomal enzymes of the sperm make penetrations through the Zona pellucida. So there is a kind of sperm receptor, which is a kind of glycoprotein in the Zona pellucida, and then the structure of the zona changes once one sperm penetrates, and then no more sperms can penetrate. So this is an important function of the Zona pellucida. Dr. Horvath also showed you these pictures to show that actually when the Zona pellucida appears, we have the so-called hatching of the blastocyst. Here you can see on day 6, when the blastocyst leaves from the Zona pellucida, it's like hatching. So this is also a revision from last semester. Then a little bit more details about these granulosa cells. So this is multiple layers of follicular epithelial cells around the oocyte, and special names for these cells. I already mentioned the corona radiata surrounding the oocyte in the tertiary follicles, and the cumulus ophores is this whole thing. Inside there are no vessels, it's epithelial cells, and later when they produce a lot of hormones, then there are the vessels growing in. And then they are surrounded by the Theca cells, Theca interna and externa. You can see the structural difference very well. Interna has so-called epithelioid reaction of the stroma cells. Stroma cells are connective tissue cells, but they almost look like epithelial cells here, so closely attached and rounded shape. So they become hormone-producing cells. So they have a lot of endoplasmatic reticulum and endoplasmic reticulum, mitochondria, lipid droplets. Why lipid droplets? Because they produce steroid hormones. So these are lipid hormones. The Theca externa is just like a little bit dense, more densely packed stroma. It's like a capsule. So it's just a capsule around the follicle. Then here we have the this liquor folliculi, this fluid, which is very rich in hormones, cytokines, growth factors. It's like a nourishment for the developing oocyte, and of course the granulosa cells. As I mentioned, many follicles begin to develop, but only one reaches full maturity. All the other follicles will degenerate. This is what we call follicular atresia. They become atretic by apoptosis. And you can, of course, find these apoptotic or atretic follicles in the slide as well. Then we reach day 14, when we have the ovulation, and what ovulates is already a secondary follicle, a secondary oocyte. You remember I mentioned that the primary oocyte finishes the first meiotic division shortly before ovulation. So what already ovulates is a secondary oocyte, and this immediately begins the second meiotic division, which then ends only at fertilization. So in most cases, second meiotic division is not ended. And here you can see, before ovulation, the so-called stigma appears on the ovary, and you can actually see that it's going to ovulate. Then there is a small bleeding, because there is a small wound on the surface at ovulation, so there will be a small bleeding afterwards. And what remains here, when this whole follicular fluid is swept out with the oocyte, then what remains, of course, is the granulosa and the Theca cells. They remain inside, and this is what is going to turn into corpus luteum. So here we have the corpus luteum. You see the fluid with the oocyte leaves, and then we have the granulosa and the Theca cells remaining inside. This is what develops into the corpus luteum. Luteum means yellow, so this is the yellow body, because it is indeed yellow. Why? Because it produces lipid hormones, and it's huge. Progesterone, lipid, it's actually fat. That's why it's yellow. And the fate of the corpus luteum can be, of course, two different things: when there is fertilization and when there is no fertilization. LH is responsible for the development of the corpus luteum, which you remember has a peak, so then decreases immediately. So if there is no LH, then corpus luteum will not be able to survive. So in a normal cycle, in a 28-day-old cycle, the LH is not enough after this peak to maintain the corpus luteum, so the corpus luteum will degenerate. If there is fertilization, the trophoblast cells produce a hormone, so it's produced by the embryo, blastocyst first, then embryo, the human choriogonadotrope hormone, or HCG. This is produced by the embryo telling the mother that I'm here, so telling about the fertilization, successful fertilization. So we need the corpus luteum to be maintained. And HCG is a similar hormone to LH. In structure, they are very similar. So it helps maintaining the corpus luteum. So during pregnancy, first 4 months, corpus luteum is very important. And this is our slide. Here you can see the corpus luteum is huge. As you have seen here, corpus luteum is a big structure. And here we have the granulosa and the Theca cells growing really big. These are called the granulosa lutein and the Theca lutein cells. But these derive from the granulosa and the Theca cells of the follicles. And if you go to higher magnification, you can see that the cytoplasm is really is filled with lipid droplets, is really pale because of the lipid. So they produce lipid hormones. The Theca cells also produce these hormones. And if you go to higher magnification, you can see that they are smaller cells, but the cytoplasm is also pale. with small magnification, they look like a darker layer, but please don't say that the cytoplasm stains basophilic, because we hear that a lot in the exam. If you go to high magnification, you can see that both cells are very pale. And this, in the slide, you can see that there are, you can discover corpus luteum from previous cycles. And when it becomes a scar tissue, then you can just see a white scar, connective tissue part. This is the corpus albicans. Corpus albicans, albus means white. So this is our slide. And you will discuss in class the details and how many different stages you can find, but actually you can find different, all the stages that we discussed here. You can see primary, secondary, these are secondary, then tertiary, secondary follicles, bigger and smaller, with the Zona pellucida, with the Granulosa and the Theca cells. And then here you can, with this magnification too, you can see the tertiary follicles because they are really big. And in some slides you can find part of the uterine tube covering the ovary. This here is the fimbria of the fallopian tube. It can be found in some slides. Here you can find atretic follicles. Atretic follicles are the degenerating one, looking as if the whole thing would collapse. And here you can see that this doesn't look normal. So this is a degenerating or atretic follicle. Please don't forget that our corpus luteum slide is also from the ovary. So here you have to talk about the whole ovarian cycle, all the other follicles, the follicular development, the hormones, not only the corpus luteum. Okay, in the slide you can find sometimes other follicles and the stroma around as well. So this is just a summary of the cycle. As I said, please practice it. And from every direction, you should know where each hormone is produced, which hormone acts on it, and the effect, the main effect of that hormone, etc. Okay, then just a few clinical remarks at the end of the ovary topic. Polycystic ovary, I'm sure a lot of you heard about it. You have seen how big these follicles can be and can become. And this is, of course, a disease when a lot of these appear, and this is this usually leads to a lot of problems and infertility. And everybody heard about the in vitro fertilization, and you also had discussed this topic in first semester embryology. So let's see our second slide, which is a very easy slide in compared to the ovary. So we can do it in a much shorter time, the fallopian or uterine tube. Here you will see it in macroscopical anatomy that it's covered totally by peritoneum. So it's an intraperitoneal organ, which is called mesosalpinx, this double layer that goes to the fallopian tube. Salpinx means tube, so it's the same word in Greek. So mesosalpinx is this double-layered peritoneum going to the fallopian tube and then covering it. So outside you can find mesothelium because of the peritoneal covering. And of course this is where from ovulation the development to a morula stage happens, and the fertilized egg is transferred to the uterus. The first, you will learn the parts in anatomy, but you have heard about these little fimbriae, these little processes that surround the ovary, and actually you can find very cool videos on YouTube how they wander during ovulation, and they kind of get stuck like a vacuum on top of the ovary at the time of ovulation. So this is why most of the times the egg ovulates into the uterine tube and not somewhere else. When it happens somewhere else, then we can have extrauterine pregnancy. So this I already mentioned that it's found in our slide of the ovary as well. So this is the uterine tube. Our slides, just going back, sorry, is like here, like a cross section of the fallopian tube. So here, these are the cross sections. You can find the usual 3 layers of any organ, any tubular organ. So the mucosa muscularis and the serosa. It's an intraperitoneal organ, so it has a serous layer. And so there is nothing special about it. The muscular layer has an inner circular and outer longitudinal layer, similarly to the intestinal wall. Very, very rich blood supply, which you can see even with this magnification already. Huge blood vessels supply the uterine tube, which has clinical importance. And now we're going to talk a little bit more about the mucous layer. It's also lucky to have this as a slide because you did it in the first semester. You saw the uterine tube twice, actually. You saw it when we discussed the epithelium with the gynoecilia, and you also had the uterine tube when you did the small blood vessels. Exactly because of this very rich blood supply, this is why we chose it as a slide in the first semester to show the blood vessels. So let's see the first layer, the mucous layer, which has, of course, the epithelium and the underlying lamina propria. The epithelium is simple columnar with gynoecilia. So details see first semester. Here we can see very nicely the row of the basal body and the cilia. This was the first semester slide with iron hematoxylin when the row of the basal bodies was black, and you could see very well the cilia on top. And these cells, which kind of stick out of the epithelial surface, here you can see them, they are the so-called PEG cells. They are secretory cells. Usually they say that secretory cells which don't secrete anymore but are going to be shed off. But according to other sources, they are still active. So of course, this is a continuous thing that they are continuously shed off the surface, and then we lose them, and then new cells are coming. So these are the secretory cells, which provide nutrition for the egg during its travel towards the uterus. This is only for demonstration. When we have the uterine tube with pus staining, you can see how purple it is. Of course, this pus positive surface indicating secretory activity, which is very important for this nourishment, but also it facilitates the sperm movement during fertilization. The gynoecilia, please review it from first semester, the 9 double and the 2 simple gynoecilia, and the microtubules building the gynoecilia, the 2 double and the 9 double and the 2 single. So this 9 plus 2 structure that we always say in the first semester. And this structure is anchored in the so-called basal body, which we can see as a line, as a row of the basal bodies in our slides. This is the surface with the scanning electron microscopy. You can see the cilia and the non-ciliated PEG cells. So as you may remember, and you can see from these slides, it's very, very, very folded. It's a very folded surface. And if you have such folds of the mucous membrane, you can imagine that the big oocyte traveling there can get easily stuck, okay? Plus, you learn in anatomy that the female genital system, the female reproductive system, is open to the outside world. So through the vagina, uterus, and then the uterine tube, we can easily get so-called ascending infections. So we have common infections in this area. After infections, attachments are formed. So we have little space here anyway. And then after such infections, you may not even notice, we can have here attachment. So it means that the chance is getting even higher for the oocyte to get stuck there. So there are 2 consequences. Either this woman won't get pregnant, so infertility, because this travel is compromised. The egg does not reach the uterus. And which may be even worse and more dangerous, is that it will be embedded in the wall. The fertilized egg will be embedded in the wall of the uterine tube. It cannot travel further, so it becomes embedded. As I said, the blood supply is excellent, and the beginning the beginning of the development can be in the uterine tube wall, and this can lead to very, very severe life-threatening bleeding. Of course, no pregnancy can be till the end in the uterine wall, uterine tube wall. It's too thin for that, and this is very, very dangerous for the mother. Here, you learned this also in first semester embryo, that implantation can happen in many other places. Most commonly, ectopic pregnancy happens in the wall of the uterine tube, and here you can see a developing embryo in the fallopian tube. In 99.9% of the cases, so basically always, there will be a rupture. So if not removed and not noticed on time, in time, then there will be a wall rupture, and very severe bleedings can happen, which is really life-threatening, as you see huge blood vessels supply from the ovarian artery, from the uterine artery, supply the fallopian tube. And the last slide shows the last thing that I wanted to say. Sorry for being a little bit over time. Here, there is again embryology, but it's this semester embryology. You will learn later the development of the male and female genitalia. And you can see this duct, which is the mesonephric duct, which partially gives rise to the male genital organs, the epididymis, for example, which of course will be only rudimentary in females. And these rudimentary little ducts are called epophoron and parophoron ducts, which can be found here, embedded in the mesosalpinx, and you can see them in our slide, okay, in the mesosalpinx. Any rudimentary duct, anywhere, not only here, but anywhere, is usually important clinically because there it can be infected or inflamed, and then big cysts can be formed, and then of course that can already cause problems, like in this case. So this is our slide, and if it's found in the mesosalpinx and we make so a cross section, then this little tail part, so in our slide you can find the cross section of the uterine tube and this little tail part, and these ducts are found in this little tail part, which is the mesosalpinx. If you go to high magnification, you can find these little ducts. These are the epophoron ducts, which you have to identify in the exam. So, of course, you can find many, many, many blood vessels. Please review the first semester histology as well, because, of course, ground tissues can be, basic tissues can be asked in the exam. And here, of course, we can ask all kinds of blood vessels. Nerves are also found here. And otherwise, it's an easy slide. So this was the end, and sorry for being a little bit over time. And then we go on with the uterus in the next lecture. Thank you for your attention.